Hello everyone, welcome back to my channel. In this channel I'm going to make a series on the chapter of sexual reproduction in flowering plants. In my previous videos I have already talked about different important events like pre-fertilization changes, then the pollination event, what are the outbreeding device so that the flowers can follow the cross-pollination event. So now in this video, the pollen grains and the stigma, both of the parts are mature enough to do the fertilization event. So now let's talk about the pollen pistil interaction. Okay, so now I'm going to draw the pictures of the things and I'm going to discuss about the events, what will happen during the pollen pistil interaction. Once the pollination event has happened, this is the pollen grain which will fall on the stigma. So if say for example this is the stigma style and this is the ovary inside which there is the embryo sac which have already been formed, and inside which the egg apparatus and synergids, everything is present over there, right? So pollen grain will come and fall on the stigma. After falling on the stigma of the flower, the pollen grain will start to germinate. If this is of the same species, then the pollen grain will start to germinate. Pollen grain will start to germinate with the help of two compounds. Just remember this is very important, that there is the formation of the pectinase enzyme, so that the pollen tube which is coming outside, the pollen tube which is forming from here, so that it can grow through the length of this style, this can go down with the help of boron, and there is the presence of boron. This is very important to remember, boron, and there is the formation of pectinase enzyme to dissolve the pectins which are present in the style, So it will release the pectinase enzyme to dissolve the materials inside the style so that it can go down, because until and unless it will go down, it is never possible to reach to the egg cell where it is waiting to do the fertilization event. So in the next step, after germinating from here with cells, do you remember that I have already talked about the three cell stage of the pollen grain? So inside the pollen grain there is this type of structure, right? Let me just make it clear to you. That is, once this is happening, what is inside the pollen tube, this type of structure we can find. There is one nucleus at the tip of the pollen tube. How the pollen grain or the pollen tube will know that in which way it has to go in the downside? There comes the role of the tube nucleus. So there is one nucleus at the tip of the pollen tube which will show the direction to the pollen tube to go, that in which way you have to go. In which way you have to go and find out your ovule, you have to find out your egg. So this is the role of the tube nucleus. Just right behind the tube nucleus there are two more nuclei which are known as male nucleus. These male nucleus are very important because they are taking part in the reproduction process. So these two nucleus will go and fertilize with the egg cell. Now let's see that what will happen once this will reach to the ovary or the embryo sac. Now let me once again draw the structure of the embryo sac. This is the structure of the embryo sac, where there are three antipodal cells. There are the polar central cell which is consisting of the polar nuclei, and there are the synergids which are consisting of some filiform apparatus which are like this finger-like projections, and one big cell containing big nucleus, that is the egg cell, right? This is the egg cell. These two are the synergids. Here is the filiform apparatus. And these three are the antipodal cells. This is the polar nuclei. Now once this this pollen tube has reached to this part, what will happen? This tube nucleus will be degenerated. The tube nucleus will be degenerated, and as this is degenerating, so it will release the male gamete, okay, or the male nucleus inside the embryo sac. So what will happen after releasing the male nuclei? This male nuclei, one of the male nuclei will go and fuse with the egg, and another male nuclei will go and fuse with the polar nuclei in the central cell. Okay, so the going, that is in which way the male nucleus will go, that is provided by the filiform apparatus. So filiform apparatus will be like this, they can move like this. So by movement of the filiform apparatus, the male nuclei can understand that one of them have to go and fuse with the egg, they have to enter inside the egg apparatus, and the another will have to go in the central cell where the polar nuclei is present. Now we know that what is the chromosome number? Haploid chromosome number, that is male nucleus are having this kind of chromosome number NN, and the egg nucleus is also having N. All of them are N in nature, right? All of the nucleus are N. Here only is the 2N. These two, as because there are two nucleus, NN, 2N, and this is also the another male nucleus which is consisting of N chromosome. So what it will lead to? This type of, as the fertilization event is happening 2 times within the embryo sac. In the first fertilization, the male nucleus is going and fusing with the egg cell, and in the second fertilization, male nucleus is going and fusing with the polar nuclei. That is why this event is considered as double fertilization, which is very important and characteristic event of the angiospermic plants. Okay, so this is about the double fertilization event. So what is happening? Let me just recall it once again. The pollen grain will fall on the stigma. The stigma and style will start to release some chemical components to see that whether the pollen grain is compatible or not. After checking the compatibility of the pollen grains, the pollen grain will start to release the pectinase. With the help of the boron and the pectinase enzymes, the pollen grain will start to germinate the pollen tube. Now, inside the pollen grain, there are the two cells, generative cells and the vegetative cells. From the vegetative nucleus, that will give rise to the tube nucleus in the pollen tube, which will be present at the topmost part of the pollen tube or the tip portion of the pollen tube, and the other cells will give rise to the generative nucleus, or the generative nucleus will give rise to the male nucleus. All of them are N or haploid in nature. Now what is the role of tube nucleus? These tube nucleus as well as the cells which are present here, that will start to secrete pectinase, and that will also show the direction to the pollen tube, that in which way it has to grow. So the pollen tube will grow and move to the embryo sac, and once entering in the embryo sac, the tube nucleus will start to degenerate and it will release the male nucleus. One male nucleus will go and fuse with the egg apparatus with the help of the finger-like projections which are considered as a filiform apparatus, and the another male nucleus will go and fuse with the polar nuclei which is present in the central cell, and in this way 2 times the fertilization event is happening. That's why the event is considered as double fertilization event. Now the entrance of the pollen tube can happen from 3 sides. Okay, if we have already seen the structure, that this kind of structure is present, right? This type of structure can be present in one place, that is considered as chalaza, in the other place that is considered as micropylar end. Now the pollen tube can enter through the micropylar end. So if the pollen tube will enter through micropylar end, that can be considered as porogamy. If the pollen tube will enter through the chalazal end, then that can be considered as chalazogamy. So depending upon the pollen tube through which the pollen depending upon the pollen tube, that through which region this is entering inside the embryo sac, different names are given, like chalazogamy, porogamy. Another mode is also there, that is considered as mesogamy. Okay, so these three are very important, that from where the pollen tube is entering. If pollen tube will enter through the chalaza end, then they can be considered as chalazogamy. If they will enter through the micropilar end, then they can be considered as the porogamy. Now, double fertilization will lead to the formation. This fertilization actually is called as Syngami. Okay, this is considered as Syngami. That is fusion between the egg cell and the male nucleus. That is Syngami, which is leading to the formation of zygote, that is diploid in nature. Okay, that is leading to the formation of zygote, which is diploid in nature. And in other case, that is the male nucleus when going and fusing with the polar nucleus, they will form the triploid endosperm or primary endospermic nucleus. They will form primary endospermic nucleus or pen, and that is of triploid in nature. Very important that it is triploid in nature and zygote is diploid in nature. These two are the result of the double fertilization. And what other changes are happening inside a flower post fertilization, that is considered as post fertilization event. So about the post fertilization event, I'm going to make another video, so keep on watching my videos. And if you do like if you found this video a little bit of helpful for you, then please do not forget to like, share and subscribe to my channel. Thank you.